Chapter 23, the digestive system. In this first section, I'll discuss the functions of saliva, but I'll mention that digestion of food is not really one of its functions. Similarly, the stomach is not really a digestive organ. Removing the stomach doesn't affect digestion, but it does affect meal size. We can talk about the causes of heartburn and some of the different treatments, including antacids, or these things here called proton pump inhibitors. And we can even learn why sleeping on the left side of the body can be good for those with acid reflux. I'll introduce the topic of zymogens. These are inactive enzymes that become activated where they're needed. This concept is really important in medicine because we frequently administer drugs that do the thing that we want where we want it, but also have side effects in other parts of the body. It would be ideal to have medications that were only active in the place where we wanted, when we wanted. So understanding this concept of what is a zymogen and how do they work is pretty important. We'll also talk about the coordinated functions of the stomach and the duodenum, and we'll talk about why SSRIs might lead to weight gain. We'll be focusing on some of the functions of serotonin in the digestive system. Lastly, we'll ask, where does this prefix Pepsi come from and why is it in all of these products? To do that, I'll do some basic overview of the digestive system, talking about the layers of hollow organs. This will be relevant to all of the other hollow organs that we will be discussing this term, including the reproductive tract. We'll start then at the mouth and get food ultimately down to the stomach in this first section. The digestive system includes the alimentary canal, which is the tube that begins at our mouth and ends at our anus, as well as all of the accessory organs, including our teeth and the liver and the pancreas, and some others that produce secretions that are dumped somewhere along the way inside of the alimentary canal. The job of this system is, of course, to ingest food, then digest food, then absorb the food, or its nutrients, into our bloodstream. To execute those functions will require many layers of control. This is a rather long canal, so different parts of it are going to be active at different times. And some of those parts can be a little bit dangerous if they were active at the wrong time. For instance, I don't want my stomach producing stomach acid 24 hours a day. So we will have controls that are mechanical in nature, like stretching of the stomach or intestines. We'll have controls that rely on chemical signals, for instance, the chemicals that are found in our food. And then we will have other controls that are regulated by hormones and neurons. In fact, the enteric nervous system has 100 million neurons controlling all of the different muscular and glandular activities within the GI tract. The ENS can be regulated by the central nervous system, but the control that the CNS provides is usually as simple as speeding up or slowing down all of the processes that are coordinated by the ENS. Many of the digestive organs lie within the peritoneal cavity. That's the space in the abdominal cavity that's between the visceral peritoneum and the parietal peritoneum. This space is full of peritoneal fluid, a type of serous fluid that helps to lubricate these organs as they are moving. Connecting the visceral and parietal peritoneum is a double sheet of serous membrane called a mesentery. We had to give this a special name because this membrane does not touch the body wall, nor does it touch any of the organs. Found in here are connective tissues and blood vessels that can bring nutrients to the digestive tract, like oxygen, and bring all of the nutrients that the digestive tract absorbs away back towards the rest of the body. Some of the mesenteries get special names, like the greater omentum, which hangs off the bottom of the stomach and connects to the large intestine. It drapes over the rest of the intestines, hanging like an apron. The lesser omentum is much smaller. It connects the stomach to the liver. And we also have a mesocolon, which connects the various sections of our large intestines. The GI tract is an example of a hollow organ, and all hollow organs are made from the same basic set of instructions. If you were swallowed whole by a saber-toothed tiger, for instance, inside the lumen of its GI tract, you would first come into contact with a layer of epithelium known as the mucosa. If you could get past that, you would then run into the submucosa, which is connective tissue, then to a layer of muscular tissue, and finally to a very thin serous membrane. The mucosa is always an epithelium, but that epithelium can be simple or stratified depending on the location. In the oral cavity and anus, the epithelial layer is always a stratified squamous epithelium. It is thick for protection. But in other locations, that epithelium is a simple columnar epithelium. A simple epithelium is better for absorbing nutrients than would be a stratified one. In many portions of this simple columnar epithelium, we will find goblet cells, which produce mucus, which will help to protect the epithelial barrier, as well as to lubricate food as it passes through the GI tract. It's very important to maintain the integrity of this mucosal barrier at all times. Otherwise, digestive acids and enzymes might digest our own tissues rather than our food. It also would allow bacteria to travel from the lumen of these organs into our bloodstream if there were any gaps in the mucosal layer. These bacteria could either be ones that were on my food or ones that we generally consider beneficial bacteria that live within our colon and oral cavity. Folding of the mucosa can increase surface area in regions where absorption is really important, primarily the small intestines. Just superficial to this is a layer of connective tissue called the submucosa. This is where we will find all of the blood vessels that can provide blood to the mucosa, as well as absorb all of the nutrients from the lumen of the digestive tract. There may be a number of mucus and other types of exocrine glands found within the submucosa. Next, we have a double layer of smooth muscle tissue, collectively called the muscularis externa. There is an inner circular layer and an outer longitudinal layer. These layers are named for the direction in which the smooth muscle cells run. The last layer is the serosa, which is a simple squamous epithelium, called a mesothelium, plus a little bit of connective tissue. In some regions of the GI tract, there is no serosa. Instead, just some connective tissue called adventitia. Regions of the GI tract that are not found in the peritoneal cavity would be lined with adventitia, such as the esophagus, anus, and duodenum. Those were the 4 basic layers of hollow organs, and we will continue to see these names throughout the rest of the term, as well as an overview of the digestive system. The smooth muscle tissue of the GI tract was different from skeletal muscle in that we do not have conscious control over this smooth muscle, and in fact, the smooth muscle tissue can undergo motions without any instruction from the central nervous system. The motions of the smooth muscle tissue are controlled by neurons within the GI tract itself, which have pace setter cells, similar to the pacemaker of the heart. And these will trigger 2 types of motions, called peristalsis and segmentation. Peristalsis involves the contraction of smooth muscle cells in one section of the digestive tract, a section that contains food, and then the subsequent contraction of the longitudinal muscles. This will propel the food down to the next segment, which would undergo the same types of muscle movements, and then to the next segment and the next segment in a wave-like motion. Peristalsis, therefore, propels food down the digestive tract. Segmentation involves alternating contractions between segments. This will set up a back and forth motion. This helps to mix food with the liquid secretions produced along the GI tract. By mixing food and liquids, this will help both with digestion and absorption of nutrients. Before we move on, it's important to note that both types of waves can be happening at the same time. The same way that if two people jump into a swimming pool at the same time, both of their waves will be bouncing around the swimming pool on top of one another. Therefore, segmentation and peristalsis could be happening at the same time. I would be both mixing food back and forth and slowly pushing it in one general direction. I have here a video of a peristaltic wave in the stomach. We are looking down towards the pyloric sphincter, and any moment now, a wave of smooth muscle contraction will be very visible. Aha, here it comes. Oh, that looks weird. And we're pushing everything down towards the pylorus. And ultimately, those waves will end, and another one is going to be starting here in a moment. So those are peristaltic waves in the stomach, but these same types of muscle activities would be happening down the entire GI tract. The generation of peristaltic and segmentation waves is generated by neurons found within the GI tract that are wired up in what are called short reflexes. When food reaches one segment of the digestive tract, that will trigger these sorts of muscle activities. But when the GI tract is empty, these waves should stop. To put it another way, if you were to cut out a region of the small intestines, keep that tissue alive, and then place an object at one end of that segment that you just cut away, the intestines would be able to move that object from one end of the tube to the other end, even though it is no longer connected to the central nervous system. The neurons within the GI tract can make these motions and regulate these motions with no external input. On the other hand, the brain is capable of adjusting these motions. That would be considered a long reflex, where the central nervous system speeds up or slows down the peristaltic and segmentation waves generated by the short reflexes. Smooth muscle movements can also be regulated by short-acting hormones. When food reaches a specific portion of the GI tract, chemoreceptors in that area can detect a change. That change might be the presence of specific chemicals, an alteration in the pH, or an alteration in the osmolarity of the lumen of the GI tract. These cells could then trigger the release of short-acting hormones, such as prostaglandins and histamines. These chemicals work just in a short distance. They diffuse through the connective tissues to reach nearby smooth muscles and trigger muscle movements. We call this sort of signaling paracrine signaling. And this helps us to regulate the motility of the GI tract, making sure that the smooth muscle is only functioning when food is in that segment of the GI tract. It's rather uncomfortable to have smooth muscles functioning when the GI tract is empty. Perhaps you felt your stomach rumble when you're thinking about food, but you haven't eaten yet. Most of the neurons in the enteric nervous system secrete serotonin. In fact, most of the serotonergic neurons in the body are found in the digestive tract, which is why if we use drugs that alter serotonin levels, they frequently have side effects on the digestive system. For instance, SSRIs are a popular class of drug used to treat many conditions, and these frequently have side effects surrounding the digestive tract. Sometimes we might use these drugs to specifically affect the digestive tract. For instance, serotonergic neurons can trigger vomiting, so if we block these neurons, that can help prevent people from vomiting, for instance, as they are struggling through chemotherapy. Those were the basic muscle movements of peristalsis and segmentation, and how those muscle movements were controlled, both by local neurons, and then modulated by the central nervous system, and then by the release of short-acting hormones. So you see we have controls on top of other controls. But now let's start at the beginning of the digestive tract and work our way towards the end. Food first enters the oral cavity. One of the important functions of the oral cavity is sensory analysis. We will analyze our food for taste, temperature, texture, even its ability to cause pain, to decide whether this is something we really want to swallow or not. Before we swallow it, we will want to break that food into smaller bits. This also helps to increase the surface area for digestion, but makes swallowing easier. In addition, mixing this food with saliva helps to lubricate that food, making it much easier to swallow. Many books will talk about the limited digestion that can occur in the mouth thanks to enzymes found within our saliva, but this is not what I would consider a part of the digestive tract, nor are we actually digesting our food. It's true that there are enzymes in saliva, but I'll talk about their function in a moment. The skin that lines the oral cavity isn't called skin, but oral mucosa. It is a stratified squamous epithelium, but unlike the epidermis of our skin, this one is non-keratinized. Without the water-resistant protein, the oral cavity is much moister than the surface of our skin. This epithelium here is also under much higher mitotic activity than the rest of our skin. In fact, the gingiva, or gums, are fully replaced every 4 or 5 days in some regions. The labia is a fancy name for the lips, and the oral vestibule is the space between the labia and the teeth. This word vestibule, just like the nasal vestibule, means the first space. The tongue is a muscular organ found within the oral cavity that helps to manipulate food for chewing, as well as to help taste and otherwise analyze that food for other sensations. There are 3 major salivary glands found in the oral cavity: the parotids, submandibulars, and sublingual. Each of these produces saliva with slightly different properties, but we will be considering the function of saliva as a whole. A pair of parotid salivary glands lies within the walls of each cheek, or the buccal wall. These glands produce a more serous secretion, which means a watery secretion, that empties via the parotid duct into the oral cavity just behind the second molar. Perhaps you've known somebody who could squeeze their skeletal muscles to put pressure on these parotid salivary glands and force the saliva out this duct and forcefully eject it from the mouth in a process in medicine known as gleeking. The sublingual and submandibular salivary glands empty underneath the tongue. The submandibular glands are below the mandibles, and the sublingual below the tongue. These produce a few more glycoproteins, or mucousy secretions. It's important for saliva to both have a liquid component, which helps to moisten and lubricate food as we swallow, but the mucousy portion is also important. Otherwise, all of the saliva would just pool in the bottom of our mouth. And we want saliva coating everywhere, the sides and roof as well. And this mucus helps to make the saliva sticky so that it doesn't drain off of our upper teeth and pool below the tongue. We produce about 1 liter or so of saliva every day. Most of it is produced by the smaller submandibular glands. We don't produce any saliva during our sleep. Now, many of you have fallen asleep in a class before. Not my class, I'm sure, but in other classes, and then you've drooled all over your lecture notes. So why would you drool if you were sleeping? Well, your salivary glands weren't producing any saliva while you were asleep, but they had already produced a bunch that was being kept within the gland. And there's nothing to prevent that saliva from leaving down the ducts into your oral cavity and out onto your notes while you were sleeping. Saliva is mostly water, but it does contain a number of important electrolytes, including some that act as buffers to help maintain a relatively constant pH within the oral cavity. As I mentioned previously, the mucus proteins help to make saliva sticky so that it can coat the entire oral cavity. One very important function of saliva is to limit the growth of bacteria. These antibodies here may help kill some bacteria, but they might also help some of the good bacteria to survive. Hydrogen peroxide, which can be found in saliva, just kills bacteria. Now, our mouth is not antiseptic. We always have some bacteria living in the oral cavity, but this hydrogen peroxide helps to limit the number. There are painkillers and waste products found within saliva, as well as this hormone over here, EGF. This is why the epithelium in the oral cavity grows much faster than the epithelium of your skin. And this is very important because of all of the chewing that you do. The friction generated within your oral cavity would damage the epithelium a lot more than our skin typically gets. So it's important for regions of the oral cavity to be replaced much more quickly, especially the gums or the gingiva. And then we also have a number of enzymes produced within saliva, and all of these have the same basic function. They break down molecules found in the cell walls of viruses and bacteria. Now, it's true that salivary amylase can also break down some starches found in our foods, but it takes several minutes for this to happen to an appreciable amount. Maybe you've done that experiment as a child where you chew on a saltine cracker for a couple of minutes until it begins to taste sweet, and then you would have experienced the breakdown of starch into simple sugars that your tongue can pick up on. However, we never chew our food for 2 minutes or more, so that is not the function of this enzyme. We don't need it to break down starches. We need it to break down cell walls of viruses and bacteria. This helps to keep us from getting sick from any nasty pathogens that are on our food, and also helps to limit the growth of bacteria in our oral cavity. We like having some bacteria there, but we don't want them becoming too many bacteria. So these enzymes here, such as amylase, lysozyme, and lingual lipase, are serving an antibacterial and antiviral function. These are not digestive enzymes. And I say that because if you take away saliva, we don't see defects in digestion. We do see a number of other defects, however, with people who cannot produce enough saliva. This is known as xerostomia or hyposalivation, and it can be caused by certain drugs of abuse, like methamphetamine, as well as a number of medications. And for people who suffer from this lack of saliva, they have no problem digesting carbohydrates. That's a function of the pancreas that we'll talk about later. What they do suffer from is tooth decay, bad breath, difficulty swallowing, and lack of taste. And these were all functions that saliva was necessary for. Saliva helps to limit the growth of bacteria, and without it, bacteria can thrive within the oral cavity, causing tooth decay and bad breath. And without saliva, chemicals in our food cannot dissolve to reach the taste buds, Hence, food tends to lose its taste. And lastly, without lubrication, food is both difficult to swallow, and different parts of our oral cavity tend to stick to each other, which makes it difficult to talk. Nowhere in here does somebody suffer from a malfunction in carbohydrate metabolism. That is just not a necessary function of saliva. Just a quick review of ch23. Taste was a perception. That's something that happens up in the brain and involves multiple sensory inputs, including the activation of taste buds. This was an example of gustation, as well as the activation of nociceptors. We interpret that typically as spiciness rather than pain when it's coming from the tongue. But a lot of our sense of taste had to do with our perception of olfaction, or smell, happening up in the oral cavity. We need chemicals from our food evaporating into the air, traveling up the pharynx to reach the nasal cavity, and activating the olfactory receptors there. That's a big portion of our sense of taste. And when our nasal cavity becomes plugged up, we have difficulty tasting our food. The dorsal and lateral surfaces of the tongue are bumpy, and each of these different bumps gets a specific name. The smallest ones and most numerous are the filiform papillae, and these little bumps provide friction that helps our tongue to manipulate food for chewing. The other three contain taste buds, which give us the ability to detect gustatory sensations. The fungiform papillae are found on the dorsal surface of the tongue. These are bigger than the filiform and shaped like mushrooms. Way at the back of the tongue, towards the sulcus terminalis, are the circumvolate papillae, and these contain a large number of taste buds. And then the foliate papillae, not shown on this picture here, are off to the sides of the tongue and also contain taste buds. Taste buds were a sensory organ, meaning I had more than just neural tissue. I also had to have some epithelial cells that helped to convert chemical signals from our food, dissolved in our saliva, and transmit those to nearby dendrites that can then relay action potentials to the brain. There were 6 types of taste buds, one of which were the umami receptors. These provided a sense of a savory flavor and were strongly stimulated by the amino acid glutamate. This amino acid can be found in any protein, from plants or animals, but inside of a protein it can't work. The amino acid must first be liberated from the protein, and then once free it could bind to the umami receptor. Treating or preparing foods in certain ways can pre-digest the proteins to release glutamate molecules, or you could add this one here, monosodium glutamate. The use of MSG has been linked to adverse health effects by the popular media, but it's very clear scientifically that these claims are unfounded. MSG is not dangerous, assuming you don't use so much that you get too much sodium, affecting your blood pressure adversely. But that's exactly why MSG can be beneficial. Many elderly people suffer from hyposalivation, and with lower levels of saliva being produced, their food often loses its flavor. And one way that they will compensate for this is by adding sodium chloride. Too much sodium chloride can often exacerbate hypertension that your elderly patients may already suffer from. MSG, on the other hand, either from a bottle or from certain foods like kelp, can increase saliva production and increase the flavor of foods at much lower concentrations than sodium chloride. Therefore, giving your elderly patients access to MSG may cut down on the amount of sodium chloride that they're adding to their food and help keep their blood pressure under control. Next, I'm going to talk about the teeth. The gingiva, or gums, are ridges of the oral mucosa that must make a connection to the surface of the tooth to prevent any bacteria from getting past our barriers and into our bloodstream. The visible portion of the tooth should be entirely enamel. This is the hardest substance in the body. It is made of calcium and phosphate crystals, which is very similar to bone tissue, only it's not made by a connective tissue cell, and it does not contain collagen. Just deep to that is dentin, which is also similar to bone. It's denser than bone, but not as dense as enamel. Therefore, if any dentin becomes exposed, it decays more quickly than enamel would. Deepest is the pulp cavity, which is where the connective tissue, blood vessels, and nerves are located. The outer surface of this pulp cavity contains the odontoblasts, which can make more dentin. The cells that made the enamel disappeared when the tooth erupted, so no new enamel can be produced after the teeth erupt from the gum line. Surrounding the dentin is cementum, which is attached to periodontal ligaments, which attach the teeth to the underlying bone tissue. Here are a few pictures of how the teeth develop. You'll notice that the crowns form first, and then the roots later. When the roots begin to form, it will push the crown towards the oral cavity. The primary dentition, or baby teeth, must be exfoliated before any underlying teeth can erupt from the gum line. It's therefore very important for proper oral hygiene to begin before the adult teeth have even erupted from the gum line. The teeth are already formed, or at least the enamel is already formed, before these teeth are visible. Living within the saliva in the oral cavity are a number of bacteria and other microorganisms that we collectively call the oral microbiome. These are usually good bacteria, at least in the sense that they help prevent infections by other bacteria from taking root in the oral cavity. We don't want too many of these bacteria growing, however. Therefore, the act of swallowing saliva brings many of these bacteria for a ride down to the stomach, where they are destroyed by stomach acids. Some bacteria don't want to be destroyed by stomach acids, but the enamel is hard and smooth, and therefore very slippery. To make it easier to grab a hold of, some bacteria will produce acids, like hydrochloric acid, which can erode some of the enamel, providing a handhold for the bacteria to form a biofilm. If this happens when we swallow saliva, the bacteria stay put, and bacterial populations in the oral cavity can thrive, leading to tooth decay and halitosis. To prevent this from happening, we can neutralize those acids with fluoride ions. These can come from toothpaste and persist in our saliva for a few hours after we're done brushing our teeth. But for longer lasting protection, luckily for us, fluoride will accumulate within the enamel and other hydroxyapatite tissues in the body. That way, when bacteria try to erode the enamel, there's already fluoride ions ready to neutralize the acids that they produce. And this helps to limit tooth decay and limit bacterial growth within the oral cavity. Too much fluoride can change the color of the enamel in the teeth, making it appear whiter. This is not of medical concern and is generally only noticed by dental hygienists. This occurs more frequently in children whose formula is made with fluoridated water. It has been linked to skeletal fluorosis, but this is not something of concern in the United States, where fluoride levels are kept very low, and only in countries whose industrial waste dumping policies are very lax. Numerous studies have been done on the safety and efficacy of fluoride. It definitely does not cause any form of cancer or increase the risk of fractured bones. It does not increase the uptake of dangerous elements like lead and arsenic, and it has definitely not been used by Hitler as a mind control agent. I'm not making that up. There are people who argue against using fluoride because they think Hitler used it. The amount of fluoride that is added to drinking water supplies has been reduced in the United States when it was discovered that lower doses were just as effective, and this should help reduce any possible side effects. Nevertheless, there is controversy over its use. Fluoride is neurotoxic, but then again, a lot of metals that the body absolutely needs are also neurotoxic, including zinc, magnesium, copper, and iron. Too many of any of those ions can be toxic to neurons. A study that added even more controversy to the topic came out a few years ago, where it was found that children in China who were drinking high levels of fluoride in their drinking water had reduced IQs. Not a huge amount, but we still worry about that. Nevertheless, we should take into account the dose. Maybe you noticed that little phrase on my previous slide, the dose makes the poison. If I were to tell you, you shouldn't drink beer, it's really bad for you, it'll make you stupid, based off of studies of folks who drink 10 to 20 glasses of beer per night, would that make you think twice about having a beer with dinner? I should hope not. It's a completely different story when you're talking about 1 or 2 beers versus 10 to 20 per night. And that's effectively the doses that these researchers were looking at. It's just not even in the same ballpark as what we're discussing here in the United States. There are a few countries that once fluoridated their water, but then stopped and saw no increase in the number of cavities. This does not prove that fluoride is ineffective. It proves that universal health care is effective, and there are definitely other ways of administering fluoride other than through drinking water, ways that countries that have universal health care are adept at doing. Here in the United States, the CDC has ranked water fluoridation as one of the top 10 public health advancements, and the Kaiser Foundation still counts dental cavities as the #1 untreated chronic illness in children here. And there are an estimated 100 million children with no dental insurance, nor have access to proper dental care. Now, many of you might say, I don't care. I want to go into medicine, not into dentistry. To which I would reply, the teeth are a part of the body just like any other, and everybody should get their teeth cared for by professionals, including licensed dental hygienists and doctors. But if that doesn't sway you, then how about this? Periodontal disease increases the risk of heart disease twofold, and heart disease is our #1 killer here in the United States. So this seems like a fairly simple preventative measure, not just for taking care of the teeth, but for taking care of people's overall health. The bacteria in our oral microbiome are good for us, assuming they're in the oral cavity. But if they get into our bloodstream, through bleeding gums, for instance, they can travel to the heart, triggering an inflammatory response by our immune system, which, if that persists, can increase the risk of heart disease by damaging arterial walls. So that wraps up some of the basic functions that we found within the oral cavity. And next we're going to move down to the esophagus. Next, food will travel to the pharynx and down to the esophagus. There are a large number of oropharyngeal muscles. Two of the ones that are important to us include the uvula, which will close off the opening to the nasal cavity during swallowing, and a large number of muscles which help to raise the larynx, which forces the epiglottis to cover the glottis. This leaves only one exit for food, which is down the esophagus. The esophagus is composed of skeletal muscle superiorly and smooth muscle inferiorly. This means we have conscious control over the beginning of a swallowing motion, but then subconscious peristalsis must take over afterwards. And I really shouldn't say subconscious, I should have said reflexive. The smooth muscle can coordinate this process itself. 3 places where food might get lodged include the cricoid cartilage, at the carina of the trachea, and the entrance to the stomach, or the cardiac sphincter. When food reaches the stomach, it will stay here for a while, giving the stomach time to complete its major functions. The first is protection. The stomach will secrete hydrochloric acid and a digestive enzyme which destroys protein, and this will kill any bacteria, viruses, or other nasty organisms that may have hitched a ride on our food or may have been living in our mouth. The stomach is also a storage device. It can store a large meal and then liquefy it, but only release that liquid one little bit at a time to the small intestines. There's just not enough room in our small intestines for the meals that we typically eat all at once. The muscles of the stomach can engage in mechanical breakdown, similar to chewing up in the oral cavity. And the stomach also produces intrinsic factor, which is very important for nutrition. To aid in the mechanical breakdown of food is this third layer of smooth muscle called the oblique layer. This will help to generate more vigorous mixing of food and watery secretions within the stomach. The lining of the stomach is a simple columnar epithelium, which has numerous pits which dive deeper into the tissue. Within these pits are a number of important specialized cells. The first are parietal cells, which secrete hydrochloric acid. Chief cells can also secrete hydrochloric acid, but they also secrete the inactive enzyme pepsinogen. There are also numerous stem cells. It's important for these cells to continually divide to replace any cells that get damaged by the rather harsh environment of the stomach. The production of hydrochloric acid is primarily under the control of the hormone gastrin and by local production of histamine molecules. Connected to the gastric pits are the exocrine pyloric glands. These produce an alkaline mucus, which helps to protect the stomach lining. Inflammation, caused by damage to the stomach lining, would trigger an increase in mucus production, and therefore an increase in the amount of protection. NSAIDs, which reduce the production of prostaglandins, would lead to less mucus being produced, and therefore can increase damage to the stomach. The exocrine glands also contain a few endocrine cells, which produce the gastrin and histamines that trigger the production of hydrochloric acid. When hydrochloric acid is in full production, the pH of the stomach can drop below 2. This means that the lumen of the stomach has some of the harshest conditions of the human body, comparable only to the inside of the pancreas. To protect the stomach lining, we have that bicarbonate-rich mucus coating produced by the pyloric glands, epithelial cells, which keep all of the hydrochloric acid within the lumen of the stomach and don't allow it to leak into our connective tissues, and stem cells, which replace any of the damaged epithelial cells very quickly. In addition to hydrochloric acid, the stomach also makes pepsinogen. This is an inactive enzyme that would only be activated in the lumen of the stomach, where it could begin breaking down proteins. It is formed as a zymogen, or an inactive molecule. If the chief cells made pepsin, the active digestive enzyme, the chief cells would digest themselves. To prevent that from happening, the chief cells make the inactive molecule pepsinogen, which has an extra little bit of polypeptide filling up the active site. And that little bit of peptide is not removed until it enters the lumen of the stomach, where hydrochloric acid can cleave a vulnerable peptide bond, thus releasing the active enzyme pepsin, which can begin breaking down large proteins into smaller proteins. Sometimes this is called the beginning of the digestive process. However, we don't need this pepsin to digest proteins. What we're doing here is breaking down proteins that enzymes or bacteria would be using to try and infect us. By inactivating these proteins, we can kill viruses and bacteria before any of this food is released to the duodenum. Removal of the stomach does not reduce a person's ability to digest protein. That's the job of the pancreas and its numerous enzymes. It does, however, increase the risk of bacterial infections, like a C. diff infection. There are few viruses and bacteria that can survive in conditions where the pH is below 2. There is one, however, called H. pylori. In fact, it enjoys these conditions so much that it can trigger hydrochloric acid secretion by the chief and parietal cells in an effort to reduce any competition from other bacteria. With increased hydrochloric acid production comes an increase in the risk of damage to the stomach lining, called a gastric or peptic ulcer. This can be further worsened by stress hormones, which increase acid production, and anything that limits stomach mucus production, including aspirin. Aspirin is an acid, acetylsalicylic acid, but aspirin is less acidic than the acids produced in the stomach. The reason aspirin can harm the stomach is because it reduces the formation of prostaglandins, which in turn trigger mucus production. Now one might wonder, why don't these bacteria, if they can survive in the stomach, harm any other parts of my body? And the answer is, no other part of my body has a pH of around 2. It's way too alkaline everywhere else in my body. So even if these H. pylori make it to the duodenum, they wouldn't be able to survive there. If the cardiac sphincter does not close completely, stomach acid can leak into the esophagus and up towards the oral cavity, damaging the tissues here, because these tissues are not protected by the bicarb-rich mucus coating. To prevent this from happening, sleeping on the left side of the body or elevating the upper body can put the cardiac sphincter in a more superior position from the rest of the stomach, keeping the stomach acid in the stomach. Furthermore, we can reduce stomach acid production right before bed by not eating or drinking right before bed. Also, losing weight and avoiding certain drugs like nicotine and caffeine can help as well. There are a number of different types of bariatric surgeries, such as gastric bypass, which can reduce or eliminate the functions of the stomach. This does not reduce our ability to digest and absorb nutrients, with the exception of vitamin B12, which must first be protonated by stomach acid before we can absorb it across the intestinal lining. It does, however, remove the ability of the stomach to store food. Therefore, we can no longer eat large meals without causing pain and discomfort to the duodenum. With repeated stress to the duodenum, this could cause the duodenum to enlarge and effectively replace the stomach. Some bypass surgeries are thought of as cosmetic, and they may be done for cosmetic reasons to reduce body weight, but it is not a plastic surgery. You would not see the difference in a patient entering the clinic and leaving after the surgery. Gastric bypass surgeries can be done for medical reasons, however, as these surgeries have been shown to effectively reduce the complications of obesity, including hyperlipidemia, hypertension, and type 2 diabetes. That wraps up the major functions of the stomach and the major cell types that we see in the stomach, including the simple columnar epithelium, which keeps all the stomach acid in the stomach, the mucus cells, which protect those epithelial cells with an alkaline-rich mucus, the parietal cells, which secrete hydrochloric acid, and the chief cells, which also make pepsinogen, and then a couple of endocrine cells, which produce gastrin and histamines. The functions of the stomach are regulated by the brain, by neurons within the enteric nervous system, and by a couple of hormones made by the digestive system. We can break down the secretions of the stomach into three phases. The first is the cephalic phase. The sight and smell or thought of food can activate regions of the brain like the hypothalamus, which then, through cranial nerves, increase the secretions of the oral cavity and the stomach. This will also lead to an increase in the activity of the stomach muscles. If you're thinking about food for too long without actually eating food, you might feel the muscle activity of your stomach, causing your stomach to rumble. When food reaches the stomach, this will trigger the gastric phase. Food in the stomach can be detected by a number of different types of neurons, including stretch receptors and a few different types of chemoreceptors, which will detect a change in molecules found in the lumen, such as the presence of peptides, and a change in the pH. These cells can then trigger the release of the hormone gastrin and local release of histamines. These will trigger hydrochloric acid secretion and muscle motility. If too much hydrochloric acid is produced, this can inhibit the secretion of gastrin and histamines. The release of hydrochloric acid will help to eliminate any harmful bacteria and viruses, and this will begin to liquefy the food, which can then be safely transported to the duodenum. Peristaltic waves are generated by the smooth muscle of the stomach and regulated with reflex activity of local neurons. We saw a video of these motions earlier in the lecture, and they push food down towards the pyloric region at a rate of about 3 waves per minute. Down towards the pylorus, the peristaltic waves reduce, and instead we see more segmentation type motions, which help to mix our food with the liquid acid secretions produced by the gastric pits. This should help convert our solid food into a liquid that we call chyme that could be delivered in small amounts to the duodenum. If it's not liquid enough, the pyloric region could force this food backwards for more vigorous mixing. Not all food can be liquefied at the same rate. Carbohydrate-rich meals dissolve in the liquid secretions of the stomach more quickly and can therefore be passed to the duodenum more rapidly, whereas fatty foods take longer to dissolve in the watery secretions. Therefore, drinking alcohol with a fatty meal will lead to a slower release of alcohol to the duodenum, whereas drinking alcohol on an empty stomach, that alcohol is already liquid, so it could be passed to the duodenum very quickly, where it would all be absorbed all at once. The third phase of stomach activity is the intestinal phase. If the pyloric sphincter doesn't do its job and accidentally releases solid food to the duodenum, stretch receptors here can signal that this is a problem, triggering reverse peristalsis and also triggering the release of gastrin, which should increase stomach acid production, hopefully liquefying the food before it's passed to the duodenum again. When liquid chyme reaches the duodenum, it is detected by chemoreceptors sensing a change in the pH. These will trigger the release of hormones like secretin and CCK, which can inhibit stomach acid secretions while increasing the secretions of the duodenum and pancreas. Therefore, when acidic chyme reaches the duodenum, the stomach is in its last phase, meaning it's time for the intestines to take over. We're shutting down stomach activity. Those were the 3 phases of stomach activity, which you can think of as the beginning, middle, and end, or the cephalic, gastric, and intestinal phases. And we also covered some of the important muscle contractions in the stomach. We've got both peristalsis and segmentation occurring at different amounts at different regions of the stomach. but this would be a good spot to take a break and come back when you're ready. And welcome back. In the second section, we're going to be focusing on the digestive function of the liver, which is to produce bile. The liver has numerous other functions, enough so that we could have written a chapter entirely about the liver. I won't have time to cover all of those functions, but I will cover a few that are related to medicine. For instance, why is mixing alcohol plus drugs of abuse often lead to an overdose? The alcohol makes those drugs stronger. On the other hand, you may have to tell your patients when they're taking certain medications that they shouldn't drink alcohol. Not just at the same time, like we saw with an overdose risk, but during the same day. We say that alcohol can contraindicate certain medications, making them weaker. We'll also talk about how some of your patients, not very many, but a couple, might be ultra-rapid metabolizers. Really strong opiates may do very little for them, and yet a weaker opiate like codeine might cause an overdose. So how is it that their liver could be both stronger and weaker? We'll also talk about removal of the gallbladder and a little bit about the small intestines. We'll start with the small intestines. Their function is more straightforward, and then we'll talk about the pancreas, the liver, and the gallbladder today. We left off with the stomach releasing small amounts of acidic chyme to the small intestines. It's within the small intestines that almost all digestion and nutrient absorption occurs. The intestines are divided into 3 segments, a small duodenum, and much longer jejunum and ileum. For starters, if your patient is counting his or her calories, that will only tell them how many calories have entered the stomach. Not every molecule that enters the stomach will be digested and absorbed within the small intestines. For instance, the speed at which chyme travels through the intestines will affect how many calories or nutrients are absorbed along the way. Surface area is also an important factor, and inflammatory diseases can affect this greatly. The length of the intestines can also be an issue. That can be modified by the longitudinal muscle layer of the small intestines, as well as by surgery. And the rate of absorption is also important. Smaller and water-soluble molecules tend to be absorbed more quickly than larger lipid-soluble molecules. Acidic chyme will first reach the duodenum. This is a very short segment of the small intestines, but a very important one, because it's here that the regulation of liver and pancreas secretions is centered. We need to match the secretions of those 2 organs with the contents that have just left the stomach. Once the acidic chyme from the stomach has been neutralized and mixed with digestive enzymes from the pancreas, it will next reach the jejunum, where digestion and absorption can begin in earnest. From there, materials would travel to the ileum next. More digestion and absorption can occur here until the ileum connects to the colon via the ileocecal valve. This specifically connects the ileum to the cecum region of the colon. The small intestines has folds upon folds upon folds. The largest folds are the plicae circulares, which can be seen with the naked eye, and these are permanent foldings within the intestines. Under the microscope, we can see microscopic villi, which are finger-like projections made of lots and lots of columnar epithelial cells. If you were to zoom in on just one of those epithelial cells, you could see the microvilli, which I'm not going to bother to try and illustrate here. Microvilli are so small that we cannot see them under the light microscope. But you have already been looking at the villi, even if you didn't notice it, back when we learned about tissues in BI 231. One villi, or a villus, is easily visible under the light microscope. It is a large structure made of lots and lots of simple columnar epithelial cells. The microvilli, on the other hand, are not visible with our light microscopes. They are no more than a fuzzy border at the apical surface of those simple columnar epithelial cells. All of this folding upon folding upon folding is similar to origami. It greatly increases the surface area. 1 square centimeter of the intestines has the same surface area as this doubles tennis court over here. We can see the plicae circularis using a fairly regular camera placed at the end of a flexible tube. When that tube is inserted through the mouth, we call that upper endoscopy. And if it's inserted through the rectum, that can give us a view of the colon, or colonoscopy. But if you want to see anywhere in the middle, well, that's too many turns for a tube to take comfortably for your patient. Therefore, using a small camera inserted within a pill would be ideal. And this is called capsule endoscopy. Notice over here to the right, we can see the plicae circularis. You can't physically see the villi, but if those villi have been damaged, it would change the color and texture of the intestines. And so we could detect that sort of damage with an endoscope. The villi were finger-like projections outward. There are finger-like invaginations inwards called the crypts of Lieberkühn, or intestinal crypts. It's within here that we find a number of mucus-producing glands. In the duodenum, they would be secreting a large amount of bicarb-rich mucus to help protect the duodenum in the same way that we protected the stomach. There are also a number of endocrine cells, which produce a large number of hormones. I'll cover a couple of them as we go through the small intestines, but towards the end, I'll cover the rest. Because the microvilli look fuzzy under the microscope, we call that the brush border. And within here, we find a number of enzymes on the apical surface of the plasma membrane of these columnar epithelial cells. These enzymes break down a number of molecules, both in our food, as well as activating another enzyme called trypsinogen. Also found on the apical membranes of these cells are the CFTR proteins that we learned about in the respiratory system chapter. They're doing the same thing that they did in the lungs, pumping chloride to the lumen, and then water follows by osmosis. We need to move a lot of water to the lumen of the small intestines to help keep our chyme moist so that it travels easily. This also helps to buffer the acids that just came from the stomach, and it keeps the digestive enzymes in solution. Otherwise, they wouldn't function. More water can be moved to the lumen of the intestines if the CFTR proteins are turned on full blast. This would lead to diarrhea. For people who are heterozygous for CFTR mutations, they would have less water moving to the lumen of their intestines when they tried to turn these proteins on full blast. For this reason, cystic fibrosis mutations in people who are heterozygous for those mutations can actually protect against the disease cholera, which kills people by triggering diarrhea. On the other hand, carrying 2 defective alleles for cystic fibrosis leads to the disease cystic fibrosis, which can affect the lungs, and here we see why it can also affect the digestive system. Chyme exiting the stomach mixes with the secretions of the duodenum, pancreas, and liver, and from here it is propelled towards the jejunum by peristaltic contractions. These smooth muscle contractions are regulated by the enteric neurons in this area. We can further modulate these contractions by activity of brain centers and hormones. That was a very quick overview of the activity and anatomy of the small intestines. To understand what goes on here in more detail, we need to cover a couple of other organs. I'm going to start with the pancreas. The pancreas has 2 major functions. The first is exocrine in nature. It secretes a large number of enzymes that mix with fluids rich in bicarbonate, which helps to neutralize the acidic chyme coming from the stomach and then digest the contents of that chyme. The pancreas also has an endocrine function. We will focus on the release of insulin and glucagon in response to changes in blood glucose levels. When we eat solid food, it must first mix with acidic secretions from the stomach to become liquid chyme. This is then released to the duodenum one small amount at a time, where it can activate chemoreceptors. These duodenal cells can then release the hormone secretin, which acts on the pancreas to produce and release bicarb-rich juices containing digestive enzymes. This hormone also acts on the duodenum itself, triggering the glands to secrete bicarb-rich mucus for protection. Vagal stimulation, which means parasympathetic stimulation, can do the same thing. For the same reason that the chief cells of the stomach produced pepsinogen, which wasn't activated to form the enzyme pepsin until it reached the lumen of the stomach, the pancreas also secretes a number of inactive enzymes. For instance, trypsinogen, chymotrypsinogen, and procarboxypeptidase must be activated in the duodenum to form trypsin, chymotrypsin, and carboxypeptidase, enzymes capable of digesting proteins. Other enzymes made by the pancreas aren't as dangerous and can be released in their active form. That doesn't necessarily mean they're active right away. Often in the alkaline environment of the pancreas, these enzymes aren't active. They only work at pH 7, when the alkaline secretions of the pancreas mix with the acidic secretions of the stomach to form a neutral environment. Enzymes embedded in the brush border of the duodenal cells are key in the activation of these proteases coming from the pancreas. Trypsinogen must come into contact with brush border enzymes to become the active enzyme trypsin. And then trypsin can activate both chymotrypsinogen and procarboxypeptidase. And now these three active proteases would be able to begin digesting the proteins that have reached the intestinal lumen. Enzymes, which can break the peptide bonds found within proteins, are called proteases and peptidases. These might convert a large protein into smaller proteins, or perhaps even cleave off individual amino acids, which are going to be easier to absorb across the intestinal lining. Pancreatic alpha amylase can break down complex carbohydrates into simple sugars. Because we have another amylase up in the oral cavity, it's important to mention which one you're talking about. Pancreatic lipases can break down lipids, and nucleases can break down nucleic acids like DNA and RNA. Under the microscope, you may notice regions of connective tissue that divide the pancreas into individual compartments, called septa. The other major components of the pancreas include these acini here, which are grape-like clusters of epithelial cells that are making the digestive enzymes and watery secretions. Also found within the pancreas are other regions of endocrine cells, which collectively we call the pancreatic islets, or the islets of Langerhans. Those were the two major functions of the pancreas. It has both an endocrine function, which we'll cover in great detail in the next chapter, and the exocrine function was what was relevant to the digestive system, including the secretion of enzymes, as well as bicarb-rich juices to neutralize the acidic chyme coming from the stomach. Next, I'm going to cover the liver. It's our largest visceral organ. Remember, the skin is not part of our viscera. The liver lies predominantly within the upper right quadrant, and it performs a large number of very important tasks, some of which I will cover in this chapter, including the production of bile and the detoxification of reactive molecules in the bloodstream. In the next chapter, I'll cover its ability to regulate metabolism, and we've already learned its role in hematological regulation. It breaks down old red blood cells and recycles most of their components. The liver receives blood from two major vessels, the hepatic artery and the hepatic portal vein. After the liver has done everything that it's going to do, that blood then leaves the liver from the hepatic veins to connect to the vena cava and rejoin the general circulation. This blood should now be detoxified of most of the reactive chemicals. Any excess nutrients that we got from our diet should be stored within the liver, and any nutrients we're deficient on can be added to our blood from the liver. The liver has also removed any old red blood cells, it's made a bunch more albumin, and it's also made things called LDLs, which we'll talk about in the next chapter. The anatomy of the liver is really boring. That's because the jobs of the liver are carried out on the levels of enzymes, not tissues. Nevertheless, under the microscope, we can see that the liver is composed of a whole bunch of repeating structures that we call lobules. About 100,000 or so of those throughout the entire liver. Under the microscope, we can see that at each of the corners of these hexagon-like structures are 6 hepatic triads, and in the center we find a branch of the hepatic vein. Each hepatic triad contains a branch of the hepatic portal vein, which brings nutrient-rich blood from the intestines to the liver. This blood may not only be rich in nutrients, it may contain a bunch of toxins that were found in our food. This blood will drain from that branch towards the center of the lobule. At each triad is also a branch of the hepatic artery, which is bringing blood from the general circulatory system. This blood, too, will travel towards the center. Along the way, both of these blood supplies will be traveling past hepatocytes, which now get a chance to do their jobs, including remove excess nutrients, dump in low levels of nutrients, detoxify chemicals, and also to produce bile. This bile will travel in the opposite direction, back towards the triad, where it will enter a bile duct and ultimately travel towards the duodenum. As I mentioned, as that blood enters from the hepatic triads, it will have to travel past a whole bunch of hepatocytes before it reaches the center area, which has a branch of the hepatic vein, which could carry it away from the liver. Along the way, these liver cells will absorb a bunch of solutes and secrete other materials that our blood might need. This can adjust circulating levels of nutrients within our bloodstream. Also found scattered through here, even though you can't tell just by looking at this picture, are a number of specialized macrophages. These cells do not leave the liver, and hence we have a chance to give them their own name, called Kupffer cells. Just like any macrophage, these guys can gobble up large particulate matter and debris. For instance, recycling old red blood cells. Hepatocytes can make bile and dump it into the tiny bile ducts found in the triads. These ultimately connect to the right or left hepatic ducts, which will dump that bile down towards the duodenum. If the hepatopancreatic sphincter is closed, the bile will back up into the gallbladder where it can be stored. We're waiting for fatty chyme to reach the duodenum and trigger the release of CCK. This hormone causes the gallbladder to squeeze and the hepatopancreatic sphincter to open up, so bile is emptied into the duodenum where it can begin the process of emulsifying that fatty chyme. Bile contains a number of salts, which will make it soluble with water. It also contains pigments, such as bilirubins, which are a breakdown product of red blood cells, which give bile its yellow-greenish color, plus a bunch of lipids, including cholesterol, neutral fats, and phospholipids. Altogether, these will help to emulsify fat, which is required before we can digest those fats and make them water-soluble for absorption. Over here, we see what a bunch of fats with an emulsifier looks like, this more liquidy substance called mayonnaise, and here we see the same lipids without the emulsifier. Here's the liquid component of mayonnaise and the lipid component of mayonnaise, and they are not mixing. So that's the major function of bile, to help the lipids in our food to mix with the liquids that came from our stomach and pancreas. They do not dissolve in those liquids. They are emulsified, meaning large droplets of fat are broken down into smaller droplets, which are then suspended within the liquid component of our intestines. For instance, this large fat droplet over here does not have that much surface area, so the digestive enzymes from the pancreas would not be able to digest very much before it reached the colon. To aid in digestion, we must first emulsify this fat droplet into a bunch of smaller droplets, and that is what bile does. It helps to break down these larger molecules, macromolecules, into smaller ones. This increases the surface area, but it does not make them water soluble. An emulsification is where a bunch of small droplets are evenly distributed throughout a liquid, as compared to that previous picture where there was a bunch of fat that just sank to the bottom of that mayonnaise without the emulsifier. The production of bile was the major function of the liver that was relevant to this chapter, the digestive system. But it's just one small function of the liver. I'm not going to have time to cover every function of the liver. We already covered how the liver helps to regulate red blood cell numbers in the previous term. I'll talk about how the liver can regulate nutrient storage and metabolism, as well as waste removal, in the next chapter. But for now, I'd like to focus a little bit more on this function here, how the liver is involved in drug inactivation. Those drugs could either be drugs of abuse or medications. The molecules from any pill that we swallow or any food that we eat would be absorbed primarily within the small intestines. All of the blood vessels leaving the small intestines, large intestines, and liver ultimately form the hepatic portal vein, which travels towards the liver. The liver also receives blood from the general circulatory system. Coming off the aorta, ultimately, is this hepatic artery. Both of these will bring blood to the liver, which allows it to do its various jobs, cleaning the blood and making sure all of the nutrient levels in the blood are adequate. That might mean removing excess nutrients or dumping in nutrients when there's a deficiency. When that blood is all good, it can then leave the liver from the right and left hepatic veins, which carry that blood to the vena cava to rejoin the general circulation. Before the blood leaves, the hepatocytes will have a chance to detoxify any reactive chemicals found in the blood, such as drugs and supplements, vitamins, toxins, and even remove excess nutrients. Because all of the blood from the intestines travels to the liver first, that means the liver will have a chance to remove any toxins from our food before it reaches more delicate organs like the heart and brain. This detoxification is carried out by a large family of very important enzymes called the P450s. These enzymes act like target dummies. They allow reactive chemicals to react with them rather than react with things that I really need, like proteins and DNA and stuff. This usually will inactivate those reactive chemicals, making them much less dangerous to the human body. But on some instances, it will actually make them more reactive and activate those chemicals. I'm going to start with ethanol metabolism. Only about 10% of the ethanol we drink gets absorbed in the stomach, the rest of it in the small intestines. But no matter where it's absorbed, it travels to the hepatic portal vein and to the liver first before it gets a chance to make us drunk. Depending on how quickly this alcohol is being absorbed, the liver might be able to remove most of that alcohol from our blood before it reaches our brain. For instance, if we drank that alcohol with a large fatty meal, our stomach would empty very slowly, so only a small amount of alcohol would be absorbed from the small intestines at a time, and our liver would have ample opportunity to detoxify a significant portion of that alcohol before it reached the brain. But if we drank that same amount of alcohol on an empty stomach, it would all reach the small intestines all at once, and thus reach the liver all at once, and it could swamp the hepatocytes. Therefore, more of it would be reaching the brain, making us drunker. My metaphor for this is, well, a batter on a baseball team might have a good chance of hitting a ball if I have just one pitcher throwing one pitch at a time. But what if I was allowed to have 5 pitchers on my team, all throwing fastballs at the same time? That batter would not be able to hit all 5 of those balls, and I might get 3 strikes immediately. Of course, that batter might get 1 single and 3 strikes, so I guess that would count as an out and a single. Okay, that's a weird metaphor, but the important part is that the liver can only detox so many molecules at one time. And if we dump too many into the hepatic portal vein at once, we can't remove all of them. When the liver detoxifies alcohol, it, actually makes it into a more reactive molecule first. That ethanol is first converted into acetaldehyde, which is actually 30 times more toxic than just plain old ethanol. Luckily, acetaldehyde dehydrogenase quickly converts that into a molecule which can be dumped into the Krebs cycle and removed from the body. This, of course, can be converted into some ATP in the process, hence we say alcohol has a certain number of calories. Some people have lower levels of this acetaldehyde dehydrogenase. For instance, people of Southeast Asian descent, from Korea and China, for instance. When they drink alcohol, they will make just as much acetaldehyde as I do, but I quickly metabolize that acetaldehyde, and I'm much less likely to get a hangover from this, as are many of my friends from Korea. Damage to the liver, for instance due to cirrhosis, can also inhibit the ability of the liver to detoxify chemicals like alcohol. But what we're going to see is that before this happens, the liver can actually get better at detoxifying those chemicals. Perhaps you already know that it's a bad idea to mix alcohol and many other drugs of abuse, such as heroin. If you were to inject a small amount of heroin into the bloodstream, some of that blood would be traveling towards the liver, where the P450s in the liver would have a chance to remove it by converting it into a non-reactive molecule. Therefore, that amount of heroin might be enough to make you high, but not enough to kill you. But then a week later, you go to a party and you get drunk, and you decide, hey, I want to do that heroin again. It was really great. And you inject yourself with the same amount of heroin, and it kills you. So why did the same dose kill you that second time? Well, it's because when you were drunk, there were lots of molecules of ethanol in the bloodstream, and it takes way more molecules of ethanol to have an effect on the body than it does molecules of heroin. And this P450 enzyme can bind to more than one substrate. Perhaps you learned in your biochemistry class that enzymes bind to only one substrate, but that's not true. And in fact, the P450s are especially promiscuous. The same P450 could break down ethanol, or it could break down heroin. But it only has one active site, so it can only break down one at a time. And since there's way more ethanol, that's what it's going to be doing. So that same amount of heroin that you injected a week ago, you gave your liver a chance to break a bunch of it down before it reached the brain. Now that you have a bunch of ethanol in your system, your liver's not going to get a chance to break down that heroin. And your brain gets an effectively higher dose of that heroin, which can lead to an overdose and death. Therefore, mixing drugs and alcohol is generally not a good idea because of the way these P450 enzymes function. So if using alcohol and drugs of abuse at the same time is a bad idea because it makes those drugs of abuse more powerful, using alcohol in the same day as you use a medication can make some medications less powerful, which is also bad for different reasons. When P450s are active, this will lead to an upregulation of the P450 genes, meaning 24 hours later or so, the liver will have more of the P450 enzymes. At that time, it's going to be done metabolizing all of the alcohol. If you were to drink more alcohol, the liver would be stronger at breaking it down. You've built up a tolerance now to alcohol, thanks to having a stronger liver. But a day later, if you were to take a certain antibiotic, those P450s, which are ready to break down anything that they can, might be able to break down that antibiotic. And now, because you drank alcohol the day before, your liver is faster at metabolizing the antibiotic drug, which makes that drug less effective. And it's less than useless to take a smaller dose of an antibiotic. That not only will be less effective at removing the infection, it may actually lead to an antibiotic-resistant strain of that infection. For this reason, we say that alcohol can contraindicate certain medications. Not all. These P450 enzymes are promiscuous. They have many chemicals that they can break down, but they don't break down everything. Ethanol, therefore, can increase the levels of certain P450s. Tobacco can do the same thing, and these would make the liver too effective at removing certain medications from the bloodstream. On the other hand, chemicals found in all of these sources can decrease certain P450 levels, making the liver less effective at removing toxins from the bloodstream. Generally speaking, those P450s will convert reactive molecules in the blood into less reactive molecules, hence inactivating them. Sometimes, though, they will convert a molecule into a more reactive one, like what we saw with ethanol first being converted into acetaldehyde. We see the same thing with acetaminophen. This chemical here must first be converted into a more reactive molecule that could then be detoxified by the liver. But that will require this cofactor here, glutathione. And if you run out of this cofactor, then high levels of this reactive intermediate can build up, causing poisoning. For instance, the therapeutic index of Tylenol. This is not a number that you're going to learn about in my class, but you would in a pharmacology class, which is the lethal dose divided by the amount that you need to be effective, is roughly 2, compared to the TI of Valium, which is 100. That means it takes 100-fold more Valium above the effective dose to be a lethal dose. And yet Tylenol is over the counter, and Valium, despite being a lot safer, is not. To make matters even more complex, not everybody has the same levels of P450s. Some people can be called fast metabolizers, and an even smaller subset of those people are ultra-rapid metabolizers. If you give these people painkillers like OxyContin, they may come back and ask you for more because the dose that you gave them didn't do anything for their pain. You might suspect that this is drug-seeking behavior, but it could just be that their liver removed that drug too quickly from the bloodstream for it to have any effects on the pain. For these people, certain drugs will be ineffective, but others might actually be more effective. For instance, codeine is a very weak opiate, found in some over-the-counter medications. It doesn't do anything on its own. It must first be converted by the liver P450 enzymes into morphine, and now it can have an anti-cough or anti-pain effect. For the ultra-rapid metabolizers, they convert this codeine into morphine so quickly that it all gets converted essentially all at once, which can lead to an overdose. For everybody else, we convert this codeine into morphine much more slowly, hence we never get that much morphine in our bloodstream at any one time, simply because we took some cough syrup. On the other end of the spectrum are people that have very low levels of certain P450s. We would call these people slow metabolizers. they would get no pain relief from codeine because they can't convert it into morphine fast enough before other P450s break down that morphine into an inactive metabolite. On the other hand, morphine would be much more likely to cause an overdose in these patients because they can't metabolize that morphine quickly enough. But wait, we're not done yet. There are gender differences between certain P450s, like this one here, the CYP3A4 protein. Women tend to have higher levels of this enzyme than men, and this is one of the more important P450s, at least in medicine, because it breaks down about half of the medications that we prescribe. For that reason, even though you might want to give a female a smaller dose if she is of smaller size than your average man, you might have to increase that dose based on the fact that her liver is going to be metabolizing that drug more quickly. This particular P450 can be inhibited by a number of foods, which is why you might have to tell your patients to avoid grapefruit juice while they're taking certain medications. The P450s were a large family of enzymes which can interact with reactive molecules and convert them into less reactive or non-reactive molecules. For instance, when I eat oranges, they contain methanol, and when I eat pears, they contain formaldehyde. Those are both reactive molecules, and generally we don't consider those good for human health. Those fruits also contain a number of good things, non-reactive molecules like proteins, carbohydrates, and vitamins. All of these molecules get absorbed and travel to the liver via the hepatic portal vein, and the P450s get a chance to interact with the reactive ones, the methanol or the formaldehyde, and convert those into safer molecules before everything travels to the rest of the body. And for that reason, even though oranges and pears technically contain toxins, the dose is so small that the human body is never harmed. The liver is just too good at detoxifying the bloodstream. For everything that the liver can't handle, the kidneys can do the rest, removing everything that's not a nutrient from the bloodstream, at least things that are water soluble. So can a milkshake or smoothie, a particular diet, or a machine add anything to your health above and beyond what your liver and kidneys are doing? Well, probably not, with the one exception. If you are on a certain diet that is making you pay attention to what you eat, you might be less likely to be eating things that are bad for you. And this is definitely good for your health, but not because that diet is detoxifying chemicals. You're simply not ingesting as many toxins. Now, I can't debunk every detox regimen, but I can debunk at least one. How about these foot devices here? You put your feet in a saltwater bath, some electricity is run through it, the water turns all dark and it begins to stink, and the person who just charged you $200 or more tells you that those are toxins leaving the body. And it's no such thing. Electricity can cause the electrodes to oxidize, causing rust, which turns the water brown, and chloride ions, when you strip away electrons from them, form chlorine gas, which evaporates and stinks. In fact, you don't even need feet in these machines to cause the water to turn dark and to begin to stink. No toxins have been removed through that very thick epidermal layer. No such thing has occurred. Okay, that's enough about detox. Let's get back to the digestive function of the liver, which was to produce bile. This bile is dumped down the hepatic ducts, ultimately connecting to the duodenum, where it can emulsify food. That is, of course, if the hormone CCK is being produced. Otherwise, that bile will travel back upwards to be stored temporarily in the gallbladder. If the gallbladder is surgically removed, then that patient can no longer store large amounts of bile, and they will have problems digesting fat-rich foods because they cannot emulsify that fat first. Therefore, they would probably need to cut back on their fat intake. The liver will continue to produce bile. There just won't be any place to store it. but that will put pressure on whatever portion of the cystic duct remains, and that can cause the cystic duct to enlarge and grow. It doesn't turn into a new gallbladder per se, but because it's larger, we can now store a little bit more bile, and so gallbladder function has essentially regenerated. Because bile is composed of bile salts, the salt portion helps to make the lipid portion water soluble, but the salt portion can crystallize. So bile can turn into bile stones or gallstones. These will not travel through the bile ducts very readily and can cause damage as they get pushed downwards. This can trigger a bunch of inflammation and pain. They can be broken up either physically or by using certain drugs, like bile acid pills. For the same reason that we didn't want to produce stomach acid, except when it was needed, we don't want to be producing pancreatic secretions, except when they're needed. Only now we're at the opposite end of the pH spectrum. Those alkaline juices and digestive enzymes could do damage to the body tissues. We only want them digesting foods. What's more is I want an equal amount of acidic chyme from the stomach to mix with an equal portion of alkaline juice from the pancreas. So we're going to tightly control how much pancreatic juice is produced and when, and that control centers around the duodenum. The control of pancreatic secretions is primarily regulated by both neurons and hormones. Neural controls include neurons found within the enteric nervous system. These can engage in reflexive movements of muscle tissues, as well as pancreatic secretions. By reflexive, I mean we don't require any activity from the central nervous system. The CNS can speed up or slow down these reflexes with activity of the sympathetic and parasympathetic nervous systems. There are also a large number of hormones which come from the duodenum. The duodenum secretes these hormones depending on what type of food has just entered the duodenum from the stomach. I'll be going through a number of them one at a time. The first hormone is gastrin, which we already learned about when we talked about the stomach. This hormone is produced when food reaches the stomach, and it increases both stomach motility and secretions. When the duodenum makes this hormone, it does the same thing. It increases stomach activity. The duodenum makes gastrin when the stomach passes along components to the duodenum that are not fully liquefied. Therefore, the duodenum needs to tell the stomach to work harder next time. The next hormone is enterocrinin, and this hormone is released whenever chyme enters the small intestines here. It will trigger the production of mucus, which helps to coat the duodenum and protect it from the acidic chyme that has just left the stomach. Next up are a pair of hormones, but I'd like to focus on just this one here, glucagon-like peptide 1, both because it has a better name and because it's more clinically relevant at this time. Both of these hormones are released when chyme reaches the duodenum, and they will signal to the pancreas to release insulin. Normally, insulin is released when blood glucose is high. These hormones can tell insulin to be released before blood glucose spikes. When food reaches the duodenum, we know that blood glucose will probably be going upwards next. And if not blood glucose, then probably blood amino acids, which insulin also regulates. Next up is vasoactive intestinal peptide. Vaso always refers to blood vessels, and this hormone makes them active in the intestines. So when this hormone is released from the duodenum, more blood should be flowing through the mesenteric arteries and veins to pick up nutrients and bring them to the liver. It also stimulates the secretion of some intestinal glands while inhibiting the function of the stomach. Next up is secretin, which is produced when acidic chyme reaches the duodenum. That's pretty much any chyme that was made from solid foods being liquefied in the stomach. This hormone will trigger the release of bicarb-rich
fluids from the pancreas, as well as secretions from the liver. When fatty foods reach the duodenum, that triggers the release of the hormone cholecystokinin, or CCK. This hormone has two major functions. We already talked about how it triggers the release of bile from the liver and gallbladder, but it also activates the pancreas to release lipase enzymes, which can digest fat after it's been emulsified by the bile. It'll take about 5 hours for food to reach the large intestines. Along the way, the small intestines are constantly churning the contents to change the environment just close to the mucosal layers. This is important because as we absorb nutrients across the mucosa, it would set up a local concentration gradient, where nutrients are high in our tissues, but low just at the surface of the lumen here. So we need to constantly mix the entire contents of the lumen so that we don't get those local concentration gradients, which would inhibit our ability to absorb more nutrients by diffusion. Water-soluble nutrients will be absorbed across the epithelium and into the capillaries found within the submucosa, which ultimately connect to the mesenteric veins, which become the hepatic portal vein and travels towards the liver. Therefore, all nutrients and toxins have a chance to be regulated or detoxified by the liver before they travel to any important tissues like the brain or heart. Fat-soluble nutrients, on the other hand, must first be packaged up into water-soluble macromolecules, known as a chylomicron. We'll talk more about these lipoproteins in the next chapter. But these are large macromolecules, meaning they have lots of molecules in them, and they're too large to diffuse across endothelial cells. They must therefore enter the lacteals through the mini-valves found in those vessels, and ultimately, traveling through the lymphatic system, connect to the left brachiocephalic vein to enter our circulatory system. Under the microscope, the duodenum should have a noticeably higher level of mucus glands in the submucosal layer, and hopefully an extra thick coat of bicarb-rich mucus for protection, because the stomach was dumping acidic chyme here, and it needed to mix with alkaline juices, both of which could damage an epithelial cell before they interacted with one another. In the other portions of the intestines, we need to do away with that thick mucus coat so that we can absorb nutrients across the epithelial layer into those capillaries and lymphatic vessels. In the jejunum and ileum, therefore, we don't see as many mucus glands or thick mucus coat. And in the ileum, we see an extra protective measure in these Peyer's patches that we learned about last quarter when we learned about the immune system. That wraps up a number of major duodenal hormones. You must learn a sentence or two about each of these, including when they are released and what their targets are. We also discussed the liver and some of its major functions, and the one major function of the gallbladder. But this would be a very good time to stop and take a break and come back later when you're ready to start learning about the large intestines. Welcome back. In this third of 3 parts, we'll talk a little bit more about diarrhea. I'll also introduce this concept of bioavailability. It's one thing to eat a bunch of nutrients, but we have to be able to absorb them and use them. We'll learn how to poop. I know you've all been excited to learn that. We'll talk about endogenous gut flora and discuss which ones are good for us, which ones are bad for us, and why. And I'll also introduce this topic of the difference between an allergy versus a sensitivity. Celiac disease is mediated by the immune system, whereas gluten hypersensitivities might be mediated by the digestive system, even though the treatment for both can be the same, eliminating gluten from the diet. To do that, We'll start talking about the large intestine and work our way down towards the rectum and anus. And then we'll wrap up with an overview of digestion and absorption. Some of the major functions of the colon include the reabsorption of water and the absorption of important nutrients and vitamins made in the colon. Why do I say reabsorption of water? Well, when you drink water, you absorb most of it in the small intestines, like most nutrients. But then the small intestines also dumps a bunch of water into the lumen using the CFTR protein. I want to reabsorb that water a little bit later, here in the colon. The colon also compacts and stores our feces until such a time that we are ready to defecate. The first part of the colon is called the cecum, which is a little pouch-like segment. Off of the cecum is the appendix. We then have an ascending, transverse, and descending segments to the colon, and lastly is a sigmoidal section of the colon, which would then attach to the rectum. Materials that were not absorbed in the small intestines would first reach the cecum. Coming off of the cecum, in the opposite direction that food would be traveling, is the appendix, or the vermiform appendix if you're not into the whole brevity thing. I didn't hear about another appendix in the human body until I was in my 40s, so I think it's safe to simply call it the appendix. And this is a lymphoid organ. This is where we have white blood cells and good bacteria waiting to recolonize the colon following diarrhea. The colon itself is larger in diameter but has a thinner membrane than the small intestines. It is divided into individual haustra. These little segments can expand and contract depending on whether feces is in the colon or not. Bands of smooth muscle, called the taeniae coli, contract the colon into those haustra, but permit for expansion. The colon also has a number of mesenteries attached to it, including the greater omentum and mesocolon, and within these serous membranes are a large amount of fatty deposits, called the epiploic appendages. After the colon, materials would then reach the rectum, which is a retroperitoneal organ, which could then temporarily store that feces until it's ready for elimination out the anal canal. The anus is lined with a stratified squamous epithelium that is keratinized. This is for protection. We definitely don't want to be doing any absorption of materials here. The movement of materials out the anus is regulated by two sphincters, the internal anal sphincter and an external anal sphincter. Just like in the urinary tract, the internal sphincter was under the control of a reflex, while the external anal sphincter required input from the central nervous system. The internal anal sphincter does more than just let feces pass to reach the external anal sphincter. It detects exactly what materials want to get past. It can tell the difference between a gas, a liquid, and a solid, and send that information up to your brain, which is going to make it very different for you as to how quickly you want to reach a bathroom, or whether it's okay to open up one of those sphincters and allow some gas to pass by. Because very little absorption is happening in the large intestines, we don't need villi here. But because the materials are being compacted and we're reabsorbing water, it's going to be hard to move them unless they are lubricated. So there are a number of glands that will produce mucus to help components travel through the colon smoothly. Some materials can be digested in the colon, but not by human enzymes. These molecules would be broken down by enzymes made by the bacteria that live in the colon. For instance, they might feed off of indigestible carbohydrates like dietary fiber. To help protect us from some of these endogenous microorganisms are a large number of lymphoid nodules found within the submucosa of the colon. This helps to ensure that all of those critters stay where they're okay, here in the lumen, and don't sneak their way past the mucosa into our bloodstream. 90% of the absorption of nutrients occurred in the small intestines, so that just leaves 10% for the large intestines. But it's a very important 10%, especially the reabsorption of water, but we also recycle a number of those bile salts, and a few vitamins are produced in the colon, so this is the only place where we could absorb them. We don't send materials backwards to the small intestines. The colon reabsorbs 9 liters of water per day. Remember, a lot of this water was dumped into the small intestines through the CFTR protein activity. This helped to provide an environment for digestive enzymes to digest our food. But then we want to reabsorb this water, and that occurs here in the colon. Without this activity, this here is the amount of diarrhea that you would have every day. So that's a pretty important job for our colon to be taken care of. When we learned about the immune system, we learned that diarrhea was an effective way of cleansing the colon. Too much diarrhea, however, can lead to dehydration, and in fact this is the #1 cause of death worldwide. Luckily, there are fairly inexpensive treatments for diarrhea. They're not always easy. For instance, having access to clean water is important. But here in the United States, it's also fairly easy to buy bismuth salicylate, or the brand name Pepto-Bismol. We don't really know how this molecule works. It might coat the inner lining of the colon, making it impermeable to water. Remember, water had to move by osmosis, so if there's an impermeable barrier, it won't be going through it. Or maybe this molecule kills bacteria that could trigger diarrhea in the first place. We're just not really sure. The colon houses a very large number of bacteria and other microorganisms like fungi and protozoans that are collectively known as our endogenous gut flora. Many of these organisms don't do us harm, so our immune system permits them to thrive within the colon. Some of them actually are beneficial to the human body. For instance, we'll cover a few benefits of these endogenous gut flora. A few of them produce vitamins that we absorb and are required for health. Many of them can prevent infections by bad bacteria simply by competing with them for nutrients. And many bacteria, in order to promote their own survival, release anti-inflammatory molecules. And in fact, it's thought that our immune system has co-evolved with these endogenous gut flora. The natural state of the immune system is to be too strong, and we just assume that the right gut flora will be releasing anti-inflammatories to lower the activity of our immune system to a point where we can fight off infections but don't get a bunch of autoimmune diseases. So why doesn't our immune system wipe out these bacteria if they're covered with bacterial antigens? Well, the colonization of the colon begins shortly after birth, and the immune system decides what is self versus anti-self sometime during fetal development and into potentially the first year or two of life. So it's possible that our immune system recognizes these gut flora as self. A number of these microorganisms live in the appendix, right next to white blood cells. If diarrhea or a broad-spectrum antibiotic were to wipe out the gut flora, some of these guys here would be able to recolonize, assuming, of course, they weren't wiped out by the broad-spectrum antibiotic as well. Diarrhea won't flush them out, because the appendix is a dead-end pocket. Many of these gut flora can modify our immune system as well. This helps them to survive in what should otherwise be an inhospitable environment. They secrete chemicals that actually lower the ability of the immune system to kill them. And this is, in fact, good for us, as I mentioned on the previous slide, by lowering inflammation. It's thought, therefore, that having the right gut flora might prevent inflammatory disease like Crohn's disease or inflammatory bowel disease. It's certainly a very attractive hypothesis that's yielding some data. There are 3 important vitamins that bacteria in the colon produce as waste products. As they digest some of the materials that we can't use, they will release vitamin K, biotin, and vitamin B5 that we absorb in the colon. Because the colon is born relatively naked, without bacteria, all newborns are vitamin K deficient, so it's fairly common to give them a vitamin K injection. Vitamin K is very important in the clotting cascade, which is why many newborns, if they don't receive this injection, would be at a high risk for bleeding disorders until their gut bacteria colonize the colon and begin producing enough vitamin K. Another very important task of our gut bacteria is to break down bilirubins, one of the waste products that came from recycling red blood cells. The bilirubins are dumped into the intestines in bile. When they reach the colon, they can be further metabolized into urobilinogens, which can be reabsorbed and enter the bloodstream to be excreted in urine. Others are converted into stercobilinogens, which turn brown and stay in the feces, giving it its color. Because newborns do not have the bacteria that can do this, they cannot effectively break down all of the bilirubins that their liver is producing as it recycles the newborn's red blood cells. Therefore, many babies are born with jaundice. This is not a sign of liver failure and is normal and not harmful. But still, we can get rid of those bilirubins by hitting them with UV light. In the old days, you might have stuck your baby in a windowsill. Of course, a few babies fell out of those windowsills, so it's much safer to use one of these machines here, sometimes lovingly referred to as grow lights. Not all of the bacteria that live in our colon, digesting things that we can't digest, produced waste products that are useful to us, like vitamin K. Some bacteria will produce gases and other molecules that have particular odors, like ammonia, indoles and scatols, and hydrogen sulfide. These things then become flatus or farts. Excessive levels of these molecules can be rather embarrassing for the individual and may require alterations to diet and possibly someday alterations to the gut flora. If you give those bacteria a whole bunch of the foods that they like, they're going to produce a whole bunch of those gaseous waste products, and that gas can cause bloating and discomfort for the person involved. This is known as a food sensitivity. If we can't digest that food, and bacteria can, they can produce gases that might be uncomfortable. There might be treatments for food sensitivities. If you don't want to simply avoid the foods that those bacteria are digesting, you might be able to eat the enzymes that digest the molecules that are the problem in the first place. For instance, for people who have a sensitivity to beans, we can eat an enzyme that breaks down the carbohydrate found in those beans. And for some people, they might have an intolerance to milk. It's possible to eat the enzyme or pre-digest the lactose and instead purchase lactose-free milk in the first place. And this can help cut down on the amount of bloating and discomfort from the gases that bacteria produce. On the other end of the spectrum are food allergies. There are chemicals in certain foods that might activate a patient's immune system, and allergies must be treated differently, usually with some sort of drug that modifies the immune system. 2 examples of food allergies are celiac disease. Well, that one's a food allergy, and then Crohn's disease is an immune system related disease that affects the gut as well. Bacterial cells typically don't live for very long, and in fact over half of the dry mass of feces is usually dead bacteria and other microorganisms. If we were to kill off more bacteria, such as with the use of a broad-spectrum antibiotic, That would inhibit their symbiotic functions, like the production of vitamin K, which could lead to a temporary vitamin deficiency. It can also destroy their ability to compete with bad bacteria, which could lead to opportunistic infections, like C. diff. That, in turn, could trigger the immune system to activate diarrhea. This would be another side effect of broad-spectrum antibiotic use. It's an indirect effect. If you've taken a microbiology class, you might have discovered that C. diff is everywhere. So why don't we get C. diff infections all the time? Well, it's an opportunistic infection. It needs an opportunity, and that opportunity is usually a colon devoid of our beneficial bacteria. For that reason, one of the main treatments for C. diff infections is, after a broad-spectrum antibiotic, to give the patient a fecal transplant to try and repopulate their colon with good bacteria so that the C. diff doesn't come back. We're starting to learn more about what are good bacteria. For instance, a couple guys have been identified. For C. diff to survive, the spores need to be activated by bile acids. And this bacterium over here likes to eat those bile acids, which prevents the spores from germinating. But if those spores were able to germinate, the C. diff starts producing toxins, called toxins A and B, which can start damaging the epithelial cells, making it easier for the C. diff to get nutrients as it leaks out here into the colon. These good bacteria can gobble up nutrients, preventing the C. diff from growing, even if it was allowed to germinate in the first place. So this good bacterium is good on multiple levels, both by blocking germination and by blocking the growth of C. diff, which in turn blocks the production of the toxins that do the damage that we associate with this colon infection. After molecules have passed through the colon, we've had a chance to digest them in the small intestines. Some bacteria digested other materials in the colon, possibly producing some goodies like vitamin K that I absorbed there. I also reabsorbed a bunch of water, which dries up the feces, and the haustra helped to compact it into units that I could then defecate. The process of defecation will be very similar to the process of urination. We first trigger a reflex, but then we wait for the brain to say that defecation is okay. When materials reach the rectum, they will go past a couple of valves to hit the internal anal sphincter. This will cause relaxation of that sphincter, but then we'll wait for the brain to open up the external anal sphincter. It's not until both of those sphincters are relaxed that defecation can occur. That defecation involves peristaltic motions triggered by the smooth muscle of the rectum and anus, as well as flexing a bunch of skeletal muscles, which can help to raise the anus, as well as putting pressure on the abdominal cavity. That's the Valsalva's maneuver, which could push the feces out. If food has reached the rectum, and the internal sphincter reflexively opens up, but the external sphincter remains closed for a period of time, this will ultimately trigger reverse peristalsis, where the rectum pulls the feces back up into the colon, where more water could be reabsorbed before it was passed to the rectum again. If this occurs too many times in a row, it could lead to constipation, as the feces is too dry to be passed easily. Anything else that gets placed in the rectum and left there for a long enough period of time, like 10-15 minutes or so, will also trigger reverse peristalsis and be pulled up into the colon where it can't be easily extracted and might require a trip to the emergency department where you would get an X-ray to help the doctors remove that item from the rectum. And that's just what everybody's rectum will do if you leave something in it for too long. for whatever reason. So that was some basic anatomy and physiology of the large intestines and the rectum. Now that we've covered all of the parts of the GI tract, let's go back and do a broad overview of some of the important processes, or the physiology that was occurring along the entire route. Mechanical digestion was physically breaking up large chunks of things into smaller chunks, but we're not breaking any chemical bonds. This primarily happens in the oral cavity and the stomach, but emulsification is also a form of mechanical digestion that happens in the small intestines. This will then increase the surface area that we have for enzymes to engage in chemical digestion, where we take large molecules and break them up into a larger number of smaller molecules. This is the primary function of the pancreas, to produce all of the enzymes that engage in chemical digestion. We did, however, see some examples of where these enzymes could also be located up more anteriorly in places like the stomach and the oral cavity. But what I said was that these enzymes were engaged more in antibacterial function rather than digestive function. Once we get a large number of small enough molecules, I need to absorb them, and that primarily happened within the small intestines, although there was a little bit of important absorption happening in the colon as well. To get a molecule across an epithelial barrier, however, it's got to be fairly small, usually small enough to fit through a pore or other sort of transport protein. Really large molecules like proteins and nucleic acids wouldn't be able to do that. Hence, the pancreas serves a very important function. We covered a large number of digestive enzymes, but not all of them, and I said that they were primarily produced by the pancreas. But we also saw examples of similar proteins secreted by the stomach, the tongue, and the salivary glands. And these types of enzymes can break down large molecules into smaller ones. Carbohydrases, like salivary amylase, can break down starches into simple sugars. Proteases break down proteins into smaller proteins or possibly into individual amino acids. And lipases can break down fats into smaller molecules, like glycerides and fatty acids. Here is my summary cartoon of all of the hormones that I have covered in this lecture and where they are produced, listed in the white boxes, as well as all of the digestive enzymes that we've covered, listed in the colored boxes. There's one that I didn't get into, which is renin here, and I mainly add this to differentiate it from renin, which was the hormone that we learned about when we discussed the urinary system and the kidneys. Renin is a digestive enzyme made by the stomach which helps to break down milk proteins, causing them to curdle, and is very important in the production of cheese. So just be careful about the number of N's that you put when you are typing out renin on your lab exams and lecture exams. That wraps up the basics of digestion and absorption that we've covered. The last thing that we need to cover are a few diseases that help to illustrate some of the physiology that we've already learned. Let's start with inflammation of the intestines. Anything that causes inflammation of the small intestines, or potentially the large intestines, such as an infection, might lead to the inability of those organs to absorb nutrients properly. Therefore, a side effect of these infections, or other conditions that cause inflammation, might lead to vitamin deficiencies, which will make for rather odd symptoms. For instance, Crohn's disease is a disease where the immune system suddenly attacks bacteria containing flagella that it hadn't been attacking before. This causes inflammation of the intestines, which can lead to a lot of abdominal pain, as well as malabsorption of nutrients. Remember, we need a fairly thin epithelium to allow molecules to diffuse across them into the bloodstream. If you thicken the mucosa, that will decrease the amount of nutrients that can diffuse into our bloodstream in the given amount of time that they're going to have traveling through the intestines. Next, let's go back to that concept that there was a difference between a sensitivity versus an allergy. A hypersensitivity was caused by the digestive system, especially endogenous gut flora digesting things that we can't, producing gases that might cause discomfort and pain, whereas an allergy is mediated by the immune system. Celiac disease is an example of an allergy. The immune system, activated by gluten, can trigger inflammation. And if this continues, chronic inflammation can begin damaging these villi until they are more or less lost. This greatly reduces the surface area of the small intestines and can lead to a reduction in nutrient absorption, leading to vitamin deficiencies. Which vitamin? Well, I suppose that depends on the person's diet. So people with celiac disease might display a wide range of symptoms. But that inflammation usually leads to general GI discomfort, pain, and diarrhea, in addition to one or more vitamin deficiencies. One way to treat this is to simply eliminate all gluten from the diet. Gluten is not a molecule, but a shape of proteins, such as gliadin, which can trap air within tiny little pockets during baking. The immune system might recognize these shapes as antigenic, causing celiac disease. It's estimated that fewer than 1% of the U.S. population actually has celiac disease, or the allergy to gluten. And yet, a large number of your patients are going to be on gluten-free diets. They don't have an allergy, but they might be sensitive to foods that contain gluten. And it might not actually be the gluten that they're hypersensitive to. It might be to other molecules that are typically found in gluten-containing foods called FODMAPs. These are short-chain carbohydrates that can be converted into gas by gut flora, causing discomfort. This is not an allergy, there's no inflammation, but it still can cause pain and discomfort. Where I live, I suspect there's an even larger number of people who are neither suffering from celiac disease nor have a sensitivity to gluten, who are on gluten-free diets. Is there any harm to a gluten-free diet? Well, not that I know of. But we should be very careful about fads with diets. For instance, low-fat diets have shown to have no benefit with respect to heart disease, weight loss, or blood pressure. And yet many people have been on weird fat-related diets for the past 30 or 40 years or so. Initially, it was thought that saturated fats were bad and unsaturated were good. So many people switched to eating trans fats, a type of unsaturated fat, thinking it was going to make them healthier. And as you may already know, this did not make anybody healthier, only sicker. We'll learn more about why in the next chapter. Then people switched to low-fat diets, and those once again did not make anybody healthier. On their own, a low-fat diet might not be harmful, but you have to ask, what is that fat being replaced with? Most people weren't content to just have bad-tasting food, and so many foods had sugars added to them to compensate for the lack of fat within the food. And this has contributed to a rise in type 2 diabetes here in the United States. So here in the U.S., we don't have a really good track record when it comes to translating medical knowledge into dietary advice. And that is both true for medical doctors and dietitians. These fat fads have ended up causing more harm than good. So we should take any diet fad with a grain of salt and start asking some of the hard questions. Like, if you're eliminating something from your diet, what are you replacing it with? And is that really better for you? Lastly, let's talk about colon cancers. These cancers are frequently deadly if they're not caught early, which is why regular medical examinations are recommended for all people of a certain age. Colon cancer is the second leading cause of cancer-related deaths in males in the United States, just after lung cancer. It frequently starts off as a polyp. Not all polyps are cancerous, but if they're removed, that reduces the risk of any of them turning into colon cancer later. That's why it's recommended that everyone get a rectal exam starting at the age of 50 up until the age of 75. This is agreed upon by the National Cancer Institute, the CDC, and the U.S. Preventative Services Task Force. I'll be mentioning these folks later when we talk about other cancer screening tests, like screening for prostate cancer and breast cancer. For those cancers, there's a lot more controversy. There's just no controversy for colon cancer screening. It's been scientifically shown to be effective. So how do polyps form? Well, remember, it's important for our intestinal epithelia to maintain a continuous sheet and have no gaps. It's hard to get the exact right amount of mitosis, however. So to prevent any gaps from forming, we usually err on the side of too many cells. Then the question comes, how do you make room for extra cells if you need to? Well, this epithelium would have to bend a little bit to make room for this next cell. And if I made a few more before others died off, well, I'd have to bend outwards a little bit more. And if I kept on doing this, ultimately, I might form a polyp. This is completely normal for the colon. The colon does this just to be on the cautious side and make sure there's no gaps for blood to leak out into my feces or feces to leak into my blood. But some of these polyps might turn into colon cancer, therefore it's good to remove them if they become too big. If you let them get really big, it might cause other problems, like blocking the movement of feces through the colon. We could detect these polyps. They're soft tissue, so they won't show up on an X-ray, but if you dump a bunch of really heavy material into here, like a barium enema, the lighter tissue here would show up as a negative space on the X-ray. Another way to try and find these polyps is with digital rectal examination, when the doctor uses his or her fingers to try and feel for polyps. Or you can use a camera. But that wraps up a few diseases that illustrate some of the physiology that we learned about in previous sections. And this is the end of a fairly lengthy chapter.